Hello, everyone, and welcome to today's webinar, Benefits of Integrating Targeted Next-Generation Sequencing Panels into Molecular Diagnostics of Solid Tumors. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by QIAGEN. To learn more about our sponsor, please visit QIAGEN.com. I would now like to welcome our speakers. Speaking today, we have Dr. Michela Bulfoni, biologist, Department of Medicine, Institute of Pathology, University of Udine, Italy. Krishna Amin, Associate Director, Global Product Management, Targeted Genomics, QIAGEN. And Dr. Thirumurthi, Associate Director of Global Product Management for Oncology, QIAGEN. Now, we encourage you to participate today by submitting as many questions as you want at any time during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues you may have seeing or hearing the presentation, and we will help you directly. Just use that Q&A box. And unfortunately, today, Dr. Bulfoni is unable to join us for the live Q&A. However, if you have any questions specifically for her, please indicate that in your question, and it will be answered via the email address you provided at the time of registration. For a complete biography on our speakers, you may click that presenter tab at the top left of your screen. Michela, Krishna, Uma, thank you for joining us. You may now begin your presentation.
Good morning. Thank you very much for your time today. I'm pleased to participate and share our experience with implemented targeted next-generation sequencing for the molecular characterization of solid tumors. My presentation is very practical, but I hope that it could be an inspiration for your laboratory activities. I believe that incorporating new technologies to study tumor heterogeneity represents a significant advancement in translational medicine. It could create a more robust dialogue between laboratories and clinical settings, as well as between research and diagnostics. My presentation is divided in three main parts. First, a concise introduction describing the scenario in which our activities are inserted. Second, an overview of our methodologies and panel selection process. And third, an analysis of our results, including the number of tests conducted and the benefits derived from our new approach. But now, let's start. Let's begin with the concept of translational medicine, which involves a disease-targeted approach aimed to enhancing our understanding of disease through the characterization of biological processes and the development of new targeted therapies. Precision medicine is a bidirectional concept, from bench to the bedside, which aims to enhance the efficiency of new therapeutic strategies, and from bedside to the bench, providing feedback on the implementation of new treatments and opportunities for their improvement. Furthermore, precision medicine aims to identify patient subgroups that are homogeneous in terms of biological features, risk of progression, response to therapy, to achieve the goal of personalized medicine, one patient, one drug. The rapid evolution of precision medicine has been driven significantly by the process in omics technologies, such as next-generation sequencing and third-generation sequencing approaches. NGS allow for rapid and cost-effective analysis of DNA, RNA, and epigenetic modification at high resolution. This detailed molecular profiling helps in identifying specific genetic alterations, biomarkers, and molecular signatures that are associated with disease susceptibility, progression, and response to treatment. This advancement, coupled with sophisticated bioinformatics tools and computational analysis, facilitate the translation of genomic data into actionable clinical insights. Together, these omics technologies have revolutionized the landscape of personalized medicine by enabling clinicians to tailor treatments to individual patients based on their unique genetic profiles and molecular characteristics. This approach moves us closer to the goal of one patient, one drug, where therapies can be targeted to maximize efficacy and minimize adverse effects. NGS enables the simultaneous analysis of multiple DNA or RNA fragments and facilitates the exploration of whole genome sequencing or whole exome sequencing. This technology is characterized by two key points: sensitivity and absolute quantification. NGS is highly sensitive, capable of detecting even low levels of genetic variation or mutants within a sample. This sensitivity is crucial for identifying rare genetic alterations and may have clinical significance in disease such as cancer or genetic disorders. NGS allow for a precise quantification of DNA or RNA molecules present in the sample. This quantitative capability enables researchers and clinicians to measure the abundance of specific genetic sequencing or gene expression levels accurately. So, sensitivity and absolute quantification make DNA a powerful tool in cancer research and clinical diagnostics. It supports various applications, including identifying disease-causing mutation, monitoring treatment response, and understanding the genetic basis of complex disease. Next-generation sequencing allows to study and to identify the molecular landscape of tumors. Some key applications are genomic profiling. In fact, it allows a comprehensive analysis of cancer genomes, identifying mutation, copy number alterations, structural variants, and other genetic changes. Then we can also identify mutation detection. So, for example, single nucleotide variants, insertion and deletions, gene fusion. Then, using NGS, we can also targeted therapy selection. And so, NGS-guided molecular profiling can assist in selecting targeted therapies based on the specific genetic alteration present in a patient's tumor. Then, we can monitor treatment response. Then we can happily use NGS in liquid biopsy. In fact, NGS-based liquid biopsy analyzes cell-free DNA or circulating tumor DNA from blood samples and provides a non-invasive method to monitor tumor border, detect minimal residual disease, and track genomic evolution over time. Then we can also discover new biomarkers in terms of facilitate large-scale genomic studies to discover novel cancer biomarkers, understand cancer biology, and uncover the therapeutic targets. It also supports the translational research effort to bridge discoveries from laboratory to clinical application. Then, next-generation sequencing also helps in studying epigenetics. NGS enables the comprehensive analysis of epigenetic modification, which are chemical modification to DNA and histone proteins that regulate gene expression without altering the underlying DNA sequence. For example, DNA methylation or histone modification are important epigenetic modifications. Using NGS, we can also study transcriptomics or perform an RNA-seq profiling, or, for example, study long no-coding RNAs and microRNAs, which are very important to study gene regulation and disease pathways. The molecular segmentation of tumor refers to the process of dividing tumors into distinct molecular subtypes based on their genetic or molecular characteristics. Next-generation sequencing provides several methods that offer different information for this purpose. Each of these NGS methods offers unique insight into the molecular characteristics of tumor, helping to classify tumor into distinct subtypes based on their genetic mutation, gene expression profile, epigenetic modification, and chromatin accessibility. This molecular segmentation is crucial for understanding tumor heterogeneity, predicting patient outcome, and guiding personalized treatment approaches in oncology. So, using a single method, we can obtain different multiple information, and this information could be hollow from specimen types and different applications. The concept of clinical utility refers to the practical value of genetic or molecular information in guiding clinical decision and improving patient outcomes. In the context of genomics and precision medicine, actional variables are genetic alteration or mutation that have no potential clinical significance and can influence treatment decision or patient management. For example, variants could be useful for target therapy selection, for a prognostic and predictive intent, for clinical trial eligibility, and for the selection of personalized treatment strategies. We can also monitor response to therapy and inherit the cancer risk. So we can also including germline mutation or somatic mutation that each person carried. And so based on this mutation, we can manage cancer risk through enhanced surveillance or preventive measurements. Precision medicine requires the integration of clinical and technical skills that must be constantly updated. This integration allows healthcare providers to effectively navigate the complexity of genomic data, molecular diagnostic, and personalized treatment strategies tailored to individual patients. Furthermore, precision medicine thrives on harmonious and shared development of knowledge. Collaboration among clinicians, researchers, and technologists is essential to advance our understanding of disease mechanisms, identifying biomarkers, and develop innovative therapies. By a collaborative environment, we can also accelerate the translation of scientific discovery into clinical application that can improve patient outcome and quality of life. To keep up with the times and ensure high standards of patient health care, we have decided to implement next-generation sequencing into our routine practices. Specifically, this decision is driven by several factors. An ever-increasing need to combine histological with molecular data for accurate diagnostic classification of tumor, for example, brain tumors or endometrial cancers. In fact, integrating NGS allows us to supplement traditional histological assessment with comprehensive molecular profiling. This combined approach enhances our ability to classify tumors more precisely, leading to more targeted and effective treatment strategies. Then, providing oncologists with information on the genomic signature of tumors, prognostic and predictive markers. So, NGS provides valuable insights into the genetic mutation, copy number alteration, and other genomic features of tumor. These information help oncologists in predicting disease progression, prognosis, and select personalized therapies tailored to the molecular profile of each patient tumor. Then, need to rationalize researches in terms of cost-effective, time efficiency, allocation of resources, patient material, time, staff, and considering that no CAVD market kits are always available, we decided to implement a custom panel in our routine diagnostic. The best solution between whole genome sequencing and target next-generation sequencing approaches depending on the specific goals of the study of the clinical application. VGS is advantageous for comprehensive genomic-wide analysis and discovery, while targeted next-generation sequencing offers efficiency and depth for focused investigation into specific gene or pathway. Therefore, the choice should be based on balancing the scope of analysis, depth of coverage, cost consideration, and the clinical or research objectives. Considering all these aspects, we decided for a targeted NGS approach. In 2019, we designed and validated two custom multi-gene panels, one called the QIAGEN glioma panel for brain tumors and the other the QIAGEN routine diagnostic panel for the study of miscellaneous tumors. The first one, so the QIAGEN targeted NGS panel designed, identify 170 hotspot mutation and 35 copy number variation in key genes in gliomas. Genes are listed in the red box and are EDH1, EDH2, PDGFR-alpha, EGFR, and then other important genes for the molecular classification of brain tumors. The QIAGEN enrichment technology was based on the amplification is based on this chemistry, and it is very important for the amplification of small fragments that characterize FFPE samples. The DNA input was 10 nanograms for identify somatic variants with a 5% VAF. The panel is composed by 333 primers, and we perform an in silico validation. Sequencing was performed on the Illumina MySeq platform, then variant calling and data interpretation were performed using the CLC genomic workbench, while the tertiary analysis was conducted manually or with the QCI interpreter. Considering the second panel we designed, the targeted routine diagnostic panels identifies mutation in 20 hotspot genes in solid tumors. In particular, for endometrial cancer, for example, TP53 and POLE, colorectal cancer, BRAF, KRAS, and RAS P3 kinase, melanoma, BRAF and RAS KIT, for GIST, for thyroid carcinoma, lung cancer, pancreatic cancer, and cholangiocarcinoma. And then the chemistry is always a QIAGEN enrichment technology based on small fragments amplification. The DNA input in this case is 2 nanograms because the number of target is smaller than the other one, and the VAF is detectable is 5%, and this panel is composed by 170 primers. Also in this case we perform in silico validation, and sequencing was performed on the Illumina MySeq platform. Variant calling, so the secondary analysis was performed always using the CLC genomics workflow. As you already know, probably know, in a surgical pathology lab, FFPE, so formally fixed paraffin embedded samples, are considering the gold standard for preserving tissues. By carefully addressing these aspects during DNA extraction for FFPE samples, researcher and clinician can optimize the quality and suitable of DNA for NGS analysis, ensuring reliable and accurate genomic data for clinical and research purposes. Considering this aspect, several critical aspects need to be considered due to the challenges posed by the fixation and the embedding process. For example, fixation time and condition are very important. Sample age, extraction method, DNA quantity and quality, DNA fragmentation and site selection, PCR inhibitors, and quality control. So in this slide you can see a representation of our laboratory workflow, and then the different steps that we perform from the specimen accessioning, so from the sample to the data analysis. And then we identify genomic variants with a median coverage of 100 pair, and we consider that true positives, variants that have a VAF according to the 5%. QIAGEN targeted custom panels offer the advantages of adaptivity and customization, allowing researchers and clinicians to stay at the forefront of genetic research and clinical diagnostic by incorporating the latest genomics insight and advancement into their studies and patient care management. So the characteristics of QIAGEN custom panels is that they always keep updated, and so they are flexible, they are adaptable, they are timeless, precision and focus, and cost-effective. Considering gliomas, the WHO in 2021 proposed a new molecular classification of gliomas integrating genetic characteristics with the traditional histological parameters to provide a more precise assessment of prognosis and therapeutic options for patients with brain tumors. Key genes for the molecular classification of gliomas are the EDH mutation, the 1p19q codeletion, third promoter mutation, EGFR amplification, and the CDK2A and B homozygous deletion. The status of these different key genes allows a discrimination, the division of the classification of gliomas into distinct subtypes that are represented in the chart reported in this slide. Other molecular markers in gliomas are, for example, TP53 and ATRX mutations that are very important in order to study the genomic instability and the alteration in DNA repair mechanisms. Then the MGMT promoter methylation, which is very important because it is associated with improved response to therapies such as temozolomide in glioblastoma. And then the H3K27M mutation, which is very important in midline gliomas, particularly in pediatric patients, because it is associated with a poor prognosis. Considering endometrial cancer. As you already know, endometrial cancer is indeed the most common gynecological cancer in postmenopausal women, following the guidelines set for by the Cancer Genome Atlas, so the TCGA in 2013, we have adopted a molecular prognostic subgrouping approach. This allows us to classify endometrial cancers based on their distinct molecular profiles, providing valuable insights into prognosis and guiding personalized treatment strategies. 4 different molecular subtypes of endometrial cancer have been identified based on their distinct genetic and molecular characteristics. The first one is polyultramutated tumors. This subtype is characterized by mutation in the DNA polymerase epsilon gene, resulting in high mutation rate, so ultramutated phenotype. These tumors often have a favorable prognosis due to enhanced immune recognition and responsiveness to immunotherapy. The second group is a microsatellite instability hypermutated. These tumors exhibit defects in DNA mismatch repair, MMR, leading to microsatellite instability, MSI, and hypermutated phenotype. They are associated with Lynch syndrome and have a better prognosis overall. They are also responsive to immunocheck inhibitors. The third group is copy number low. This subtype is characterized by a relatively stable genome with low levels of copy number alteration. It includes tumor with few chromosomal aberration and mutation, often associated with endometrioid histology and a moderate prognosis. The last one, so the fourth group, is copy number high. These tumors display extensive genomic instability with numerous copy number alteration, including amplification and deletions. They are typically associated with aggressive features, serous histology, and poor prognosis. These molecular subtypes of endometrial cancer provide valuable information for clinical management, prognostication, and treatment decision-making, highlighting the importance of molecular characterization in guiding personalized therapies approach. In particular, genomic subgroups are characterized by pathogenetic mutation in the exonuclease domain of poly, corresponding to the polyultramutated phenotype, mismatch repair deficiency, so MMRD, with altered immunohistochemical expression of mismatch repair proteins corresponding to the MSI hypermutated group, then no specific molecular profile with preserved p53 and MMR immunohistochemistry expression corresponding to the copy number low group having a low mutational burden, and then p53 abnormal with aberrant p53 immunohistochemical expression, including complete loss and or overexpression of p53 corresponding to the copy number high group with a high mutational burden. In the molecular classification of endometrial cancer, key genes include poly and TP53, which play crucial roles in the tumor biology and prognosis. Poly mutations are associated with the ultramutated subtype of endometrial cancer. Poly mutation lead to a high mutational burden to the impaired proofreading during DNA replication, resulting in a favorable prognosis and potential responsiveness to immunotherapy. TP53 is a tumor suppressor gene commonly mutated in cancer, including aggressive forms of endometrial cancer. TP53 mutations are often found in the copy number high subtype and are associated with genomic instability, prognosis, and poor prognosis and resistance to standard therapies. The assessment of the mutational status of poly and TP53, along with the status of DNA mismatch repair proteins, is typically performed using immunohistochemistry surrogate test in clinical practice. And in this case, then, we can analyze the MMR proteins, so we can also identify the MMR deficiency, which is characterized by the absence of MMR protein MLH1 and 2, MSH6 and PMS2. That indicates a microsatellite instability. MMR deficient tumors are predominantly found in the hypermutated subtype of endometrial cancer and are responsive to immune checkpoint inhibitors, as I already said in the previous slide. Then, based on the information provided to now, our aims were as follows: the identification of the most common genetic variants in gliomas. Then we focus our attention on detective single nucleotide variants, insertion, deletions, and copy number variations. This approach aimed to enhance the diagnostic accuracy and provide a clearer understanding of glioma pathogenesis. Then, the characterization and classification of endometrial carcinoma for diagnosis and therapy definition. We aim to classify endometrial carcinoma based on their molecular profile, focusing on key genes like SPOLY, TP53, and MMR genes. This classification aims to guide personalized therapy decision and improve patient outcome. Then the last point is the identification of the mutational status of predictive prognostic genes in other solid tumor. In fact, this is another application of our routine diagnostic panel. In fact, our goal was to identify mutation in predictive and prognostic genes across various solid tumors. These include assessing genes such as EGFR, KRAS, BRAF, and other genes that are very important to inform treatment strategies and predict the patient outcome. All these informations have to be reported in a clinical report. For our clinical report, we follow the CIAPEC molecular and predictive pathology group guidelines for drafting NGS reports. These guidelines aim to ensure consistency, accuracy, and comprehensive information in reporting next-generation sequencing results. Key components of the report included patient information, so a double identifier, sample identification number, sample type, the percentage of cancer cells in the sample, the clinical question, so the motivation of the molecular investigation, methods employed, so nucleic acid extraction, type of NGS used, Test results in terms of synthetic results present absence of variants, and then the type of variant following the AGVS nomenclature, then a brief summary of results, the clinical significance of the identified variants, and the level of clinical significance of the variants is given according to a scale defined by Haskell-Kapp, references, and also if we participate to external quality controls. Using next-generation sequencing, the number of molecular tests in our lab has significantly increased, has doubled, reflecting advancement in genomic analysis and precision medicine. Using the custom design panel, we conduct approximately 400 tests, comprising 11% gliomas, and the remaining tests are referred to miscellaneous tumors. Among these tests, 33% were for endometrial tumors and 60% for other types of tumors. Analyzing gliomas using NGS, we observed that the frequency of EDH mutation was comparable between low-grade gliomas and high-grade gliomas, approximately 10%. Additionally, among low-grade gliomas without EDH mutation, 11% were found to have key mutation in key genes such as TERT, CDK2A and B genes, TP53, which while this figure was 10% among high-grade gliomas. So this mutation characterize high-grade glioma, which are more or less 10% of our case study of our patients. Other mutations detected in our study included variants that are reported in this table. This table could indicate the extent of heterogeneity with that sample analyzed. So glioma is a very heterogeneous, and in particular glioblastoma is a very heterogeneous brain tumor subtype. The group of Vernac et al. Proposed a molecular classification of glioma based on comprehensive genomic analysis, aiming to refine diagnosis and guide treatment strategies. Their classification system categorized glioma into different subtypes based on genetic alteration and molecular profiles, such as mutation in EDH, TP53, ATRX, and TERT promoter, as well as pattern of chromosomal copy number alteration. This approach has provided insight into the heterogeneity of gliomas and their prognostic implication, paving the way for personalized medicine in the management of these challenging brain tumors. Using this classification, we can classify our gliomas, as you can see in the graph. So 31% no mesenchymal subtypes were found, 47% classic, 9% proneural, among the 12% neural, and 31% could be classified as undefined GBM that showing an overlapping signature belonging to different subtypes. Mutation analysis of endometrial cancer contribute to their molecular classification to improving patient outcomes. The majority of endometrial cancer can be classified into one of the 4 molecular subgroups. However, in a small subset of patients, more or less 5%, the molecular analysis will show more than one classifying alteration, so they could be defined as multiple classifier. Then 10% of cases was polymutated, 11% P53 abnormal tumors, MMR hypermutated tumors more or less 28%, and then 5% NS-MP tumors, so copy number low. The mutation analysis contributes significantly to the molecular classification of endometrial cancer that enhance diagnostic accuracy, guide treatment decision, and predict patient outcome. In conclusion, NGS molecular analysis of tumor has enabled us to achieve several critical advancements in cancer management. First, deliver more accurate diagnosis by identifying genetic alteration with high precision. NGS helps in providing more accurate and comprehensive diagnostics of tumors, leading to more targeted and effective treatment strategies. Then helps us in differentiate subclones, not only driver mutations. In fact, NGS allows for the detection of both driver mutation and subclonal mutation within tumors. This capability is crucial for understanding tumor heterogeneity and evolution, which can influence treatment decision and patient outcomes. Then, NGS implementation in the clinical routine could eliminate the need for ordering multiple tests, so it is time and cost effective. So consolidating multiple genetic tests into a single NGS assay saves time and resources, streamlining the diagnostic process and reducing costs associated with traditional sequential testing methods. Last one, it allows to create a multidisciplinary management of patients. In fact, the comprehensive molecular profiles obtained from NGS facilitate multidisciplinary collaboration among oncologists, pathologists, and other healthcare figures such as lab technicians or biologists. This collaborative approach ensures the treatment plans are tailored to each patient's unique genetic profiles and clinical characteristics. In summary, NGS molecular analysis represents a transformative technology in oncology, offering enhanced diagnostic accuracy, detailed molecular insights into tumor biology, cost-effective testing, and facilitating a personalized multidisciplinary approach to patient care. Next steps are focusing on update customized panels already in use and design a new targeted DNA panel for prostate cancer. Then I want to thank all my collaborators from supervisor to colleagues for their support and expertise. Thanks to QIAGEN for their technical assistance, and then to each of you, thank you for your attention and for engagement today.
Thank you, Dr. Bulfoni, for that great presentation and nicely lining up QIAGEN targeted DNA Pro panels for me. Hi, everyone. My name is Krishna Amin, and I'm the Global Product Manager responsible for the Targeted DNA-seq portfolio for library preparation solutions at QIAGEN. And today, together with my colleague Thirumurthi, I would like to highlight how QIAGEN can help you accelerate your time to results for your NGS oncology testing. Before I get started, I would like to disclose our legal disclaimer. QIAGEN products that I will show you are intended for the molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For the agenda, we have the complete sample to insight overview for you that starts with an overview of the QIAGEN targeted DNA Pro panels for library preparation and some performance data. I will then give you a very brief process laid out in QCI secondary analysis with Lightspeed. I will then hand over to my colleague Uma to provide you with an overview of QCI Interpret for oncology. So getting started, QIAGEN Targeted DNA Pro offers a robust library preparation solution enriching targets of interest from as little as 10 nanograms of DNA. The workflow features advanced chemistry that replaces bead cleanup steps with enzymatic cleanups after the adapter ligation and target enrichment that not only cuts down your hands-on time, but it also increases your UMI capture efficiency. So the QIAGEN Targeted DNA Pro, by cutting down hands-on time, it gets you from sample to sequencing ready library in just under 6 hours with just 1.5 hours of hands-on time. When we talked about the increased UMI capture efficiency, this also helps with the high sensitivity in detecting low variant allele frequency, as we'll see in the next slides. This ensures accurate and comprehensive results. As we learned from Dr. Bulfoni's presentation, the combined assessment of IDH1 mutation status and LOH or co-deletion for 1P and 19Q is important for molecular classification of gliomas. So in this slide, I will show the feasibility of this combined assessment with minimal sample input with QIAGEN targeted DNA probe workflow. We used two different cell lines to assess the IDH1 mutation and LOH, frankly, because we could not find a cell line that had these genotypic events present at the same time. So we used the BT88 DNA and serial diluted with Na24385 DNA to obtain different tumor purities percentages. The DNA input amount into the workflow was 10 nanograms for all dilutions, and we used 5 nanograms of the BT54 into the workflow directly. We also use the QIAGEN Targeted DNA Pro Comprehensive Cancer Research Panel. This provides relative enrichment of chromosome arms of 1P and 19Q, as well as the full coding region coverage for all of the genes that are targeted by the panel. The libraries were sequenced on Illumina NextSeq platform, and the analysis was performed using QIAGEN CLC Genomics Workbench. As you can see table on the top, we can detect LOH for 1P and 19Q at tumor purities as low as 60%. And as you can see in the figure below, we're also successful in detecting the IDH1 hotspot mutation at the residue 132. So QIAGEN Targeted DNA Pro also enables high sensitivity in detecting FFP DNA variants. This is due to the increased UMI capture efficiency that you now have because of the enzymatic cleanup steps. So to demonstrate this, we use 30 nanograms of the Horizon OncoSpan FFP DNA reference standards. We prepared the libraries using the QIAGEN Targeted DNA Pro Comprehensive Cancer Research Panels. The libraries were sequenced on Illumina NextSeq platform, and the data was analyzed using QIAGEN CLC Genomics Workbench. So as you can see in the table here, we're able to detect all of the variants at the expected frequency. So this ranges from 1% variant allele frequencies all the way to 90% variant allele frequencies. Increased UMI capture efficiency also enables high sensitivity in detecting cfDNA variants. So to demonstrate that, we use 30 nanograms of the Sera-seq ctDNA mutation mix that has variants at 0.5% allele frequency. We use the QIAGEN Targeted DNA Pro Comprehensive Cancer Focus Panel that looks at the hotspots and actionable variants in the targeted genes. The libraries were sequenced on Illumina NextSeq platform, and the data was analyzed using the QIAGEN CLC Genomics Workbench. As you can see on the table here, we're able to detect almost all variants at the expected variant allele frequencies. The variants that are not detected require some pipeline optimization for detection. Our catalog has 18 QIAGEN targeted DNA Pro panels featuring both cancer-specific options and comprehensive cancer panels. These panels facilitate comprehensive genomic profiling, including immuno-oncology biomarker analysis like TMB and MSI. The panels deliver robust specificity with an impressive on-target rate of 90%, as you can see here, and uniformity of coverage exceeding 99%, as you can see here. This ensures that sequencing efforts are focused on the intended targets with minimal off-target effects and resulting in high-quality data that we saw earlier that are suitable for accurate and comprehensive genomic analysis. QIAGEN Targeted DNA Pro also offers flexibility to accommodate new content without compromising on the quality of the data produced from them, as we saw in Dr. Bulfoni's work. So there are two ways of creating a custom panel. One way is through GeneGlobe NGS Custom Builder, which allows you to create your own custom panel with their default primer design parameters. Another way is to work with our Enterprise Genomic Solutions or EGS team. The EGS team can provide with advanced customization options to target non-coding regions, as well as detecting complex variants that go beyond your SNPs and indels. The team also works with customers to ensure that the custom panel design is optimized to your NGS workflow that takes in even the sample multiplexing requirement into the consideration when designing a custom panel. So the complete QIAGEN sample to insight procedure begins with DNA extraction with QIAamp kits that are suitable to your samples. Next is library preparation and target enrichment with QIAseq targeted DNA pro panels and platform-specific indexes. Libraries that are generated can then be sequenced on a sequencer of your choice. Data can be performed using our commercial-grade QIAGEN CLC Genomics Workbench or qCI secondary analysis, which I'll go over in the next slide. The detected variants can then be interpreted with QIAGEN Clinical Insight Interpret for Oncology. So this now brings me to the next step in the sample to insight workflow, which is our secondary analysis. So when it comes to secondary analysis, in addition to CLC Genomics Workbench, we also have a cloud-based solution through QCI secondary analysis. This is where we can automate all your manual steps, so all you have to focus on is generating high-quality reports. So the raw FASTQ files can be uploaded directly to QCI secondary analysis, either from a local server or from a cloud-based platform, for example, Illumina's BaseBase. After this, the entire secondary analysis pipeline can be deployed in the cloud, and the relevant outputs, such as the VCS and QC reports, can be downloaded for review. Also, we can upload the VCF files directly to QCI Interpret, where they will be available immediately for interpretation and reporting. With that, I will now hand over to my colleague Uma to walk you through the next and the last step in the sample to insight workflow. She will walk you through the QCI Interpret for Oncology. Thank you. Over to you, Uma.
Thank you, Krishna. This now brings me to the next step in the sample to insight workflow, QCI Interpret 1 for Oncology. Legal disclaimer before we get started. QCI Interpret 1 is an evidence-based decision support software, and it is not intended to be used as a primary diagnostic tool. QIAGEN Clinical Insights, or QCI Interpret for Oncology, is a decision support software that combines automation and manual curation to deliver augmented molecular intelligence. What that means is that the software accesses a large scientific knowledge base of both manually curated and NLP-derived information to help labs to quickly and confidently prioritize and assess variants, classify them according to the ACMG and AMP guidelines, and hence interpret variants at scale. The software also pre-selects relevant variants, matches the molecular profile with therapies and clinical trials, so you can produce oncologist-ready and patient-tailored reports in a time-efficient manner. QCI Interpret has processed over 4 million cases so far and counting. QCI Interpret 1 is powered by the QIAGEN knowledge base that contains a world-leading combination of genomic and clinical knowledge. The QIAGEN KB has constantly been growing for more than 20 years now and continues to do so. It hosts information curated by our team of PhD scientists, engineers, and ontologists. The QIAGEN KB team utilizes both artificial intelligence and natural language processing, or NLP, to help identify and prioritize literature for analysis by scientists, who then read these papers and extract and curate information from text, figures, and supplements, and use standardized curation protocols to store data in a structured format. The knowledge base includes comprehensive information from drug labels such as the FDA and EMA, and clinical practice guidelines such as NCCN and ESMO. The knowledge base also incorporates data from over 25 different public databases such as Cosmic, Nomad, among many others. In the next few slides, I'll show you how QCI Interpret 1 can enable you to confidently interpret variants in your NGS workflow and deliver reports in just 3 easy steps. These are the 3 general steps in your variant interpretation workflow within QCI Interpret 1. The first is to automatically prioritize and pre-select clinically relevant variants, therapies, and trials for reporting. The second is to review, modify, or confirm variant classification and evidence. The third and the final step is to generate customizable PDF or XML reports that contain expert summarized variant and disease-specific interpretive comments. Let's begin by looking at how you can prioritize clinically relevant variants in QCI. This step is fully automated within QCI. Once you upload your VCF, user-defined pre-filters are applied to quickly prioritize clinically relevant variants. Each variant that passes through the filter cascade is then classified according to the ACMG and AMP guidelines, utilizing the vast, high-quality curated content of the QIAGEN knowledge base. Additionally, each variant is also matched with relevant treatments and clinical trials, all of which are also manually curated. This ensures that you receive accurate and best available treatment and trial matching for your reports. The software also automates the selection of variants, treatments, and trials that are included in the clinical report. With this automation in place, you can now quickly proceed to step 2, which is to review, modify, and confirm the variant classification and evidence. QCI Interpret 1 does a great job of bringing all the evidence needed for you to confidently assess and evaluate the variants in one single place, so that you don't have to spend time looking for all the information and you can be confident that you're not missing anything. The variant details page that you're looking at right now contains various information, including what contributes to the computer actionability and pathogenicity classification based on AMP ASCO CAP and ACMG guidelines, respectively, along with the relevant references that you can get to with easy clicks. On this page, you'll also have access to treatments and trials that are pre-selected for you. You are able to see the references and also have complete control to modify as per your needs. Moreover, you also have access to manually curated summary along with relevant literature written by QIAGEN scientific experts on the functional impact of variants, along with information gathered from several other resources, including curated literature from the QIAGEN knowledge base and from public databases, in silico prediction tools, etc. With all this information at your fingertips, you can now confidently assess variants and relevant information associated with them. Once that's done, we can head to the third and the final step in the process, which is to generate the clinical report. The end result from QCI Interpret 1 is a fully customizable report with relevant variant information, including ACMG pathogenicity, AMP actionability, relevant drugs and clinical trials, prognostic information, and customizable interpretive comments. The interpretive comments are written by a team of MD, PhD experts and are oncologist reviewed and contain a wealth of information about the variant. This slide shows you the information that you would get from expert written oncologist reviewed expert interpretive comments that enables you to build a comprehensive report within a matter of minutes. You'll get expert interpretation summary written by dedicated scientists, fully backed up with references, expert classification based on AMP guidelines, and the expert interpretation content is available for all variant types and biomarkers, again accompanied by relevant references that you can access easily with a click of a button. You also get variant and disease level clinical evidence that encompasses therapeutic, prognostic, and diagnostic significance that is reviewed and approved by a team of practicing medical oncologists. Moreover, information on the significance of co-occurring mutations in the context of therapeutics, prognostics, or diagnostics is also captured in the expert interpretation summaries. We've heard feedback from several of our customers that writing such comprehensive variant-level comments is a time-consuming and resource-intensive task. Now, with these pre-written variant summaries, we have already done the heavy lifting for you, And all you have to do is to review the information, select what you want to include in your reports, and hence decrease the turnaround time and increase efficiency. The QCI team is constantly working on adding new enhancements and features catering to our customer needs and also to keep up with the latest trends in the field. On these lines, the latest release of QCI Interpret 1 has several new features that are focused on enabling users to scale up with ease and efficiency, such as flagging of co-occurring variants and interactions within the app, features that enable faster test review and streamlined test prioritization, bulk assessment of variants that enables you to rapidly triage and hence save time, and many more features. In summary, with QCI Interpret for Oncology, we do all the heavy lifting so you can focus on generating the most comprehensive, high-quality reports with ease and with efficiency. And just to try everything together, the whole sample to insight workflow allows you to confidently offer high quality NGS testing in a time and resource efficient manner. Thank you for your attention, and we'll be happy to take any questions.
Thank you, Michela, Krishna, and Uma for your excellent presentations. It is now time for our live Q&A portion of our webinar. And so I'd like to welcome back Krishna and Uma for our live Q&A. We will now start it. And if you haven't had a chance to submit any questions, please do so now. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So we are coming close to our end, so we're going to squeeze as many in as possible. So let's see. Let's start with this one. Krishna, I'm going to start with you. Let's see. So does this work on Thermo and MGI platforms? Let's get you unmuted. There we go.
Yes. We saw a lot of data that was generated on Illumina platforms, but the chemistry or QIAGEN panels does work on Thermo. We've generated extensive amount of data on Thermo platforms, not just Thermo and Illumina, but also with so many new sequencers that are on the market. We've also have generated data on some of the complete genomics or MGI instruments, as well as elements of VD sequencer as well. So the panels themselves are platform agnostic and does work on non-Illumina platforms as well.
Excellent. Thank you. Uma, I'm going to jump over to you on this one. How frequently is the QIAGEN KB updated?
That's a great question. We actually have content updated every 2 weeks. So, we capture the recent approvals or guideline recommendations, and we stay, on track with the latest advancements in the field.
Thank you. Krishna, I'm going to jump over to you now. How many reads are required to detect variants at 1% and 5% VAF?
That's a very loaded question, but a great one. So the amount of reads that are required for different variant allele frequency usually is a function of how much DNA you input into the library prep workflow, as well as your panel size. So for example, if you're trying to detect 1% variant allele frequency from say 10 nanograms of DNA, and your the required read pairs per UMI is 4. So we've done some calculations for you that amounts to around 25,000x coverage. Now this is going to depend on the size of your panel as well. On an Illumina sequencer, so that might mean I think 15 to 20 million reads if you have a very relatively small panel. If you're working with the panel as large as say TMB, which has around 1.2 megabases or off-the-shelf panels, you might need over 100 million reads to get to 1% variant allele frequencies. For 5%, the number of reads required is far less. The average coverage we would recommend is 7200x coverage to get to 5% variant allele frequencies. Of course, these are our conservative recommendations. We always recommend that the customers always start with their conservative recommendations and then fine-tune the read requirements as per their lab workflow. Because many times customer will see that they don't need as much as we recommend. But of course, by our recommendations, we'll always want to make sure that you have enough reads to be able to call the variant allele frequencies that you're targeting. I hope that answered that question.
I believe it did. And I'm going to stick with you on this next question. What are the major differences between QIAGEN targeted DNA pro panels and, QIAGEN targeted DNA pro panels and QIAGEN targeted DNA panels?
So for those of you who don't know, the QIAGEN targeted DNA panels were launched in 2016, I believe. So these panels have been on the market for a number of years, and these are the panels, as Dr. Bulfoni mentions, they've been using in their lab since 2019. So over the years, we received some feedback from our customers that, the panels work great, especially when it came to FFPE samples, as we saw in the presentations, but the turnaround time really needed to be reduced. The workflow really needed to be streamlined. So what we've done with the pro panels is that we've essentially just streamlined the workflow. So the major differences is instead of doing the bead cleanup steps, we've introduced enzymatic cleanup, so that cuts down your hands-on time drastically. We've also incorporated some fast cycling enzymes that reduces your overall turnaround time. So we're cutting our turnaround time by half. Another major difference that we've done is that we've removed the need to have custom sequencing primers. So this means that you can now sequence libraries from different panels that are QIAGEN and non-QIAGEN on the same flow cell. The workflow is also now much more automation friendly than it was before because we've really re-engineered some of the enzymes to be more automation compatible and reduce the viscosity of some of the reagents. So the difference in performance might not be as much, but in terms of the just overall workflow, the pro panels offer a much more streamlined and fast workflow.
Thank you for that answer. Uma, let's jump over to you. Can you, let's see. Okay, let's do this question. Uma, what type of tumor samples can QCI interpret one process?
So QCI can process both solid and hematological tumors, pediatric cancers. So it's like anything under the sun, we can process in QCI.
Excellent. And we have time for one last question. And Uma, I'm going to target it at you. Can you select clinical trials in QCI based on the patient's location? Where do the trials come from?
Yes, absolutely. Yes, when you upload your VCFs into QCI, either through the user interface or through the API, you have an option to specify either the physician zip code or the patient zip code. And based on that, QCI will select trials up to a 500-mile radius from that inputted zip code. So you have the full flexibility to restrict the clinical trials that you want to look at. And clinical trials are coming from clinicaltrials.gov. We take the data, we curate it considering the inclusion and the exclusion criteria and other specifications in the trials.
Very good. Thank you both for this excellent Q&A session. So this concludes our session today. Krishna, Uma, do you have any final comments for our audience?
Thank you from my end, thank you for being with us for today's presentations. And if you have any questions, feel free to reach out to us.
Yeah, I just want to thank the audience for their time. And yeah, as Krishna mentioned, do reach out to us. You have the information for any additional information that you might need. Yeah.
Excellent. Well, a big thank you again to Michela, Krishna, and Uma for their time today and their important research. We also want to thank LabRoots and our sponsor, QIAGEN, for sponsoring today's educational webcast. Now, before we go, I do want to thank our audience for joining us as well and for their interesting questions. Questions we did not have time for today, and there were quite a few. And then additionally, those submitted during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Thanks again, friends, for joining us. That's all for now. Have a great day. Goodbye, everyone.
Thank you.
Thank you.